We started off by looking at the way or the structure of the genetic material in a bacterial cell, acknowledging that it is secular, like a circle, isn't it? Now, to start replication, it just doesn't start from anywhere. It must start from somewhere. And we said that the region, the position, the segment, the location of that point where DNA replication starts on the bacterial chromosome, that circular loop, is referred to as the origin of replication. The origin of replication, sometimes just referred to as the ORIC, is the position, the region that is found on the bacterial genetic material or the chromosome where replication must start. In this case, DNA replication must start. And we said it has got some features that allow the replication machinery to identify it. What I'm trying to say is that if you look at the origin of replication, it's a double-stranded segment. It also has the same nucleotides like anywhere else on the bacterial chromosome. So if it contains the same nucleotides, there must be some features that make it be recognized as a starting point. And we said that it has got two regions. It has got a region that is referred to as a 9-mer and another region that is referred to as the 13-mer. Now, some of you may be wondering why I'm going through what I said yesterday, because I got some, questions here and there. So I thought of just giving you some kind of a recap so that we can get to where we are today and hopefully we can move together. So for those of us who've understood, very good. And for those of us who are asking questions, I'm hoping that this can help you understand and help you read like I advised. So I was trying to say that the region on the DNA, the ORIC, has certain features that allow the replication machinery, and by that I mean the proteins and the enzymes that help in DNA replication identified. It has a region that is referred to as the 9th mer and another region that is referred to as the 13th mer. Collectively, these are referred to as the origin of replication. Those are found in that region. So to start replication, there are these initiator proteins that are referred to as DNA. So in this case, you're going to write DNA. The D will be in capital letters, the NA will be small letters, and then A will be capital letter. So DNA is an initiator protein. And by initiator protein, it's like that person who initiates a conversation. So think of DNA as the protein that initiates replication. What does it do? It first of all recognizes the 9 mer. How is it able to recognize the 9 mer? Because within the 9 mer, it has a binding site. And by that, I mean a region where it can bind. Okay? The first thing in this process is to identify the oriC. How is it done? We have proteins that can initiate these. These are referred to as DNA. So DNA in prokaryotes identifies the 9 mer because within the 9 mer, there is what is referred to as a DNA box, just a region where this protein can bind. Now, remember, the DNA is a double-stranded molecule held by hydrogen bonds. So there is kind of affinity between the two strands. The moment another partner comes in, and in this case, the DNA, it kind of weakens that region because not only is that DNA segment binding to its fellow DNA, but it's now binding to something else. And because of that conformational change, it kind of causes a distortion, like a strain, that allows the region in the 13 mer to just open up a bit, not very long, maybe about 10 to 20 nucleotides, okay? It's a very short segment. Now, we can't say that DNA is an enzyme that breaks hydrogen bonds. No, it doesn't do that. Here, the understanding is DNA, the double strands are stable when they're on their own. The moment you add another molecule there, it kind of becomes heavy, okay? And because in the DNA box, there's this high affinity to bind DNA, the DNA molecule in that position won't only be binding to its fellow template molecule, but another guy. So it kind of causes a strain, which allows that section to open up, to unwind. So what I'm trying to say here is that DNA is not an enzyme that, breaks hydrogen bonds, no. The breaking of hydrogen bonds is because of that strain that it causes by binding to DNA. Now, once there is that short segment of DNA that is unwound, and by that I mean the two strands in that region are no longer held by hydrogen bonds, okay? We know the 13 mer is rich in 80 bases. And this is because it is easy to open up those bases or break hydrogen bonds because we know that between an A and a T, there are only 2 hydrogen bonds. You can imagine if it was rich in G's and C's, it will mean the cell has to use more energy to break the hydrogen bonds. But like I always say, life in itself is made to conserve energy. So now, once that region has been opened, okay, we have got a region where we have got 2 single-stranded DNA molecules. It's a region, not the whole loop. Think of it just as a segment where there's 2 single-stranded, DNA molecules. Now, to continue breaking those hydrogen bonds, an enzyme comes in, and this enzyme is helicase. In bacteria, it is also referred to as DNA B. Okay? Helicase, or DNA B, I'll just refer to it as helicase, it's easy for me to say that, is the enzyme that actively breaks hydrogen bonds, separating the double-stranded DNA molecule into single strands. And it does it consecutively. It won't do it at one time. It will be doing it sequentially to allow for synthesis to take place, but not, opening a very long stretch that makes the DNA, intertwine. No, you want to just open a short segment to allow for synthesis to take place. Now, even though helicase can break the hydrogen bonds, and remember I said it uses ATP, energy. What that means is that it's an energy demanding process. It requires you to input energy. So now, once helicase has broken the hydrogen bonds, remember, these hydrogen bonds can still complementary base pair because that's how they were able to hold each other in the first place. So other proteins come in to stabilize this single strand so that they can remain single-stranded. And these proteins we said are single-stranded binding proteins, right? Now, the role of the single-stranded binding proteins, first and most importantly, is to bind single strands. Like I said, single-stranded binding proteins do not bind double-stranded DNA. They'll bind single-stranded DNA, and the reason is you want to stabilize them so that they remain single-stranded, they don't complementary base pair, because the reason why you have got that single-stranded region is to allow for each of those DNA molecules to serve as a template. So you want to keep them like that. In addition to that, when these strands are single, they can be exposed to so many things that are found in the cell. So some of these SSBs, they also protect the DNA from nucleases. These are just enzymes that can break DNA. So in short, the most important function of the SSBs is just to stabilize the strand to remain single-stranded, because for us to replicate DNA, we need a single-stranded DNA segment. Now, one more thing that happens when the DNA is open is that the helices up in front of it, it causes this supercoiling. You can imagine you're holding a rope you're holding a rope that has got two ropes intertwined. When you open a short segment, it won't be very difficult, but the more you pull, you're going to create strain up front. Now, this supercoiling, or should I say strain, that is caused by unwinding of DNA can stop DNA replication because it's going to be like pushing back on this region that you've unwound. Now, to relax these regions where there is this strain due to unwinding, we have other enzymes that come in. These are referred to as topoisomerases, or sometimes DNA gyrase. The reason why we have these guys is to ensure that the unwinding process does not create supercoiling strain that is going to make our replication bubble, or that region that we've unwound, collapse as into a new back. Yeah, I can't remember what else I've left there. Now, I think now we're at the point where we're talking about replication. So just to now, that was initiation. Now replication, or elongation, should I say, is a process where now we start adding nucleotides to this DNA. We've opened it up, it's been stabilized, supercoiling has been taken care of, so we can start adding nucleotides. In elongation, the enzyme that adds new nucleotides, to form a new DNA strand or just a daughter strand is DNA polymerase. To be specific, DNA polymerase 3. DNA polymerase 3 is the major polymerase in prokaryotes that synthesizes DNA. And by synthesis, I mean adding nucleotides to make a new DNA molecule. Now, DNA polymerase 3 has got certain characteristics. One of them is that to add new nucleotides to the growing chain, it can only add in the 5 to 3 prime direction. It can only add a new nucleotide from the 5 to 3 prime direction. What this means is that it needs a free 3 prime OH group to be sitting already on the DNA template. Synthesis is in the 5 to 3 prime direction, and I think I explained this very well. If you look at the orientation of the nucleotide, you can see that on one end there's a 3 prime. Look at the pentose sugar, where there's I think on carbon number 3, and then the 5 prime is on carbon number 5. You're going to see the phosphate. So now it can only add nucleotides in the 5 to 3, meaning that synthesis, when we're using DNA polymerase, is always in the 5 to 3 prime direction. Now, because of this need for a free 3 prime OH group, we need to synthesize a primer. And I explained what a primer is. I'm lucky this morning I'm in the house. I don't need to put all that stuff on my face, but ladies will understand. A primer in DNA is a short segment of nucleotides that provides a free, and when I say free, you know what I mean, free 3 prime OH group that DNA polymerase 3 can use to join a new nucleotide. The primer is RNA in nature, meaning that it is not made from deoxyribonucleotides. It is made from ribonucleotides, and it is synthesized by an RNA polymerase. To be specific, the RNA polymerase that synthesizes a primer is referred to as a primase. The RNA polymerase that synthesizes a primer is referred to as a primase. Now, unlike DNA polymerase 3, it does not need to have a free 3' OH group. No, it can start synthesis. And it's important for us to note that the synthesis of the RNA primer is also in the 5 to 3' direction. I want to emphasize that when you look at the RNA polymerase primase and the polymerase 3, they have one feature which is common. They all synthesize in the 5 to 3' direction. Only that one of them, the polymerase 3, needs a free OH group, while the primase doesn't need it. That is why I can save that row. So now this is where we are today. Now I want us to pay attention to this slide. Okay, let me use the next one because I decided to blow it. So at this point, we have a double-stranded stretch up front. You can see the topoisomerase is doing their job and coiling to ensure that there's no strain on this region. As you're unwinding, the more you unwind, the more supercoiling you have. So the topoisomerases come in to relax. Sometimes they even cut or twist and do all that kind of stuff. So the point I want you to focus on is this region. I have designated this strand, if you can see my pointer, as strand A, and designated this strand as my strand B. And now at this point, I want us to look at the orientation. Strand A has a 3'. This is a 3' end. Automatically, what this means is that the other strand will be a 5' end, because we know that DNA strands are anti-parallel. One runs in the 5 to 3, the other one in the 3 to 5. And also now, I want us to look at the primase. Remember, we're saying the primase is the enzyme. It is an RNA polymerase enzyme that synthesizes the RNA primer. So what we see here is a primer. And at the bottom here, we also see a primer. So then, on strand A, we've got a primer. On strand B, we have a primer. Now, I said that RNA polymerase, the primase, the direction of synthesis is also in the 5 to 3. Okay, so let's look at strand A. Keeping in mind, synthesis must be from the 5 to 3, meaning you're moving from the 5 to 3' end. Okay, so if we look at strand A, synthesis will start from here, going in this direction. If we look at strand A, synthesis will start from this end, going this end, meaning that the strand of DNA that is carrying the 3' end is allowing for us to synthesize in the 5 to 3. The same direction as this we know is a replication fork, isn't it? So here I want us to pay attention. Look at the strand. The first thing that you must be able to do is to designate the 5 and the 3' end to know synthesis of this primer. It will be for strand A, it will be from this end to that end. If this is a 5, if this is a 3', the primer end will be a 5' here. So it's synthesizing in the 5 to 3'. Now let's look at the other strand. On the other strand, we have the 5' end here. And if we're going to synthesize our primer in the 5 to 3, we can't move in this direction. What this means is that the end of the primer here will also be 5. So meaning that on this end, we must synthesize in the opposite direction. Meaning that synthesis will start from here, going this direction, away from the replication fork. It is easy for me to use a fork because we know if the replication fork is here, the direction is going that way. So what this means is that the primers that are synthesized during DNA replication, one of them is synthesized in the same direction as the replication fork, and one of the primers in the opposite direction to the replication fork. This is just like a reference point. So the take-home message from here is that the primase synthesizes the primer in the 5 to 3' direction. What this means is one of the strands, the primer will be synthesized in the same direction as the replication fork. On the other strand, the primer will be synthesized in the opposite direction to the replication fork. Now, if this is clear, I want now to move on to the features of elongation. The features of elongation at this point, we have already synthesized our primer. We understand what the primer is and what its nature, its RNA, and everything else about it. So now we want to start synthesizing our DNA. Our polymerase now has, the 3' end. So we know that polymerase 3 synthesizes new DNA by adding nucleotides to the free OH end of the primer. What we're going to see in the next slides, we're going to see that we're going to have what is referred to as the leading strand synthesized continuously in the same direction as the replication fork, and we're going to have the lagging strand synthesized in the opposite direction discontinuously away from the replication fork. So in elongation, we're going to see words, or should I say terms, such as a leading strand, the lagging strand, and also Okazaki fragments. And these ones, you must pay attention to the reference point. If you see what I did in the previous slide, my reference point was the replication fork. Either synthesis in the same direction as the fork, we're going to refer to that as leading strand. When synthesis is in the opposite direction, the replication fork, we're going to call that the lagging strand, or it is called the lagging strand. We know that elongation uses primers. This part we've already looked at it. And also, DNA replication is not immune to mistakes, meaning that in life, making mistakes is very normal. If you don't make mistakes, then you're not living. So here on point number 3 is acknowledging the fact that DNA replication is not immune to accidents or mistakes. And these ones must be corrected. And this correction is performed by DNA polymerase 3 using what is referred to as a 3 to 5' exonuclease activity. I'll explain this later. For now, maybe we can just leave it there. Okay. And also, we know this process of elongation is also coordinated by the helicase, because we need the hydrogen bonds to be unwound. We need a single template. The SSBs, these ones help stabilize the DNA. So now, let's move on to the next slide. This is kind of like the overview of what is happening at the origin of replication. Okay, and we know the origin of replication will be at this junction here, and then replication will take place going in this direction, because we know we are going to have helicase here, and also we're going to have helicase going in that direction. And I hope you remember what I said about helicase. The enzyme helicase loads on the strand that is facing the replication fork. That is to say, the helicase moves in the 5 to 3' direction. So of the two strands, you must be able to pick which one of them is going to load the helicase. Of course, sometimes diagrams on the internet, they mislead us, but the helicase always loads on the lagging strand. Okay, the strand that is in the 5 to 3' direction, because the enzyme itself in nature has polarity. It must move in the 5 to 3' direction. I hope I have a slide to explain that. So anyway, let me go back to this slide. So let's take this position as our origin of replication. What this means is that an origin of replication is going to have 2 replication forks. And by that I mean you have this region where helicase is unwinding, forming a Y, that's a replication fork. And also, we also have this region where there's a Y, meaning the helicase is opening the DNA going that way. So meaning that at each origin of replication, we're going to have 2 replication forks. At each origin of replication, we're going to have 2 replication forks. And we know that the collection of everything there is referred to as a replication bubble. a replication bubble has got 2 replication forks and 1 origin of replication. Now, I want to segment this diagram into 2 so that I can explain. So I'm going to start by showing you a region that is only comprising of this end, and then I'll show a region comprising this end, because maybe explaining everything at once might be a bit confusing. So I decided to add another slide where I cut this diagram, okay, into 2. The way I'm cutting it is on the black line, because we know replication will take place going this direction and in that direction. So maybe let's start with how replication takes place going only in this direction. So this is a slide. I've just cut the previous diagram in half at the replication of origin. So in this diagram, replication is going in this direction, the fork, the direction of the replication fork. Let's label our strands. We have got strand A and strand B. Now, of the 2 strands, one of them will be synthesized in the same direction as the replication fork, and the other strand must be synthesized discontinuously away from the replication fork. And we know why this is so. This is because DNA polymerase 3 can only add DNA in the 5 to 3' direction. So let's focus on strand A. Strand A, this end is a 3' end. If you go down on the blue line for the strand, this end is a 5' end. Strand B, if you cut it here, this end still maintains the 5' end, and this end maintains the 3' end. We can start with strand A. Synthesis must take place from the 5 to 3' direction. So then, we're going to assume there must be a small primer there, okay? So if the primer is synthesized at this junction, it's going to offer us a free OH group, and we can synthesize going continuously in this direction. So the strand of DNA that is synthesized in the same direction as a replication fork is referred to as the leading strand. And if you pay attention, you're going to see that the strand that is synthesized with the strand in the 3' is what is referred to as the leading strand. Even if I didn't know what this strand was, I'll just look at strand A and its orientation. If on this end there's a 3' end, then this new DNA will have a 5' end, and synthesis should be from the 5 to 3. Then I know this is the leading strand. Either way, you must be able to identify which one is the leading strand. At this junction, this is a 3' end, meaning I can have a new strand moving from the 5 to the 3. This strand is synthesized in the same direction as my replication fork. Okay, this is referred to as the leading strand. Let's now move to strand B. Strand B, if we cut it if we cut the replication origin in the middle, this position here becomes the 5' end. Okay, so meaning that the orientation of strand B is 5 to 3. But then, if polymerase can only add a nucleotide to a free OH 3' OH group, it means that I can't synthesize it in the same direction as replication fork. It has to be in the opposite direction. So what this means is that the primase goes up front, synthesizes a short primer. It's not properly indicated here. Let's just take it like it's a green thing. Synthesizes a short primer, and then of course pulls away to allow now the polymerase to synthesize in the 5 to 3. When it reaches this junction, it's going to fall off and assemble on another primer and synthesize in the 5 to 3. So this synthesis is going to be discontinuous until the whole molecule is synthesized. On strand B, how do I know that this is my lagging strand? The first thing I'm looking at is just its orientation. It's a 5' end, meaning that I can't have 5 and 5. DNA must always be antiparallel. So if this is a 5' end, this must be a 3' end. So meaning that synthesis can only start from this junction, 5 to 3. So in a similar way, the lagging strand also needs a primer And synthesis by DNA polymerase 3 will also take place from the 5 to 3' direction. Because this synthesis is in opposite direction to the strand, these strands are referred to as the lagging strand. And one of the differences you're going to see between the leading strand and the lagging strand is that the lagging strand is going to have multiple primers, while the leading strand only needs one primer to start, and it will continue all day because it is continuous. So sometimes the leading strand synthesis is referred to as continuous synthesis because it only needs one primer to start, while the lagging strand sometimes is referred to as discontinuous because it needs more than one primer. Its synthesis is away from the replication fork, while the leading strand, the synthesis is in the same direction as the replication fork. I hope this is clear. We can take, so in this case, I hope when you look at this diagram, you are aware that the part I was looking at is only this end. So we also have this other end. So in short, this diagram wasn't properly thing. I think it was, errors of picking diagrams from the internet, because one of these must be lagging strand, but I'll show it better. What I'm trying to say is that the part that I was talking about is only this end of the replication fork. But then we also know replication is taking place this other side. So how is it taking place? In short, the part that I've explained is only this end. We also need to understand how synthesis is taking place here. Also, we need to show which one is the leading strand, which one is the lagging strand. And again, I'm going to split this diagram into two. But of course, when you're analyzing it, you must be able to picture it as a whole, not the way I'm doing it. I'm just splitting the diagram to make it easy for me to explain, or maybe for you to follow, whichever one. Okay, so yeah, so this is the leading strand, and this one is going to be the lagging strand. So let's look at the other part of that replication of the origin of replication. So then, if this strand has got a 5' end there, that's a blue strand, this end will be a 3'. This end is a 3'. Its end should be a 5', meaning that at the replication of origin, the moment you split it, you're going to have 5, 3, and all those, designations that are going to come about. So you must be able to picture this, that on one end, if this is the 3', the other end that was cut should be a 3' end. If this is a 3, the other end should be a 5. So now, let's start with strand A. Strand A, at this point, this end is 5'. Direction of replication is in this direction. This is where the helicase is going, or this is the replication fork. If you look at strand B, this end is the 3'. Why? Because DNA is antiparallel. So if strand A is 5', this guy here should be 3'. What is the direction of the replication fork? It is going this way. So then, don't think of replication starting from here. No, they're only looking at the point where the two strands are unwound, because DNA replication only uses a single template, only one strand at a time. So we can't start reading from this end. No, you must start reading from the region that is unwound. So the first thing that I did is to establish which strand is a 5', which strand is the 3'. The next thing that I did was to check which one is the direction of the replication fork. So at this point, I'm very clear it is going in this direction. Then the next thing I'm going to look at is DNA polymerase 3. It can only synthesize in the 5 to 3' direction. So I'm going to inspect strand A. Strand A has got a 5'. Can I start synthesizing in the 5 to 3? No, because it will be 5', 5'. But we know very well to say they must be antiparallel anyway, or the enzyme itself can only synthesize in the 5 to 3. Because of that, this strand should be synthesized discontinuously, meaning the synthesis will start in the opposite direction. So there will be a primer and then synthesis this way. It won't be this continuous. Of course, we have, in fact, the Okazaki fragments, the short stretches of DNA synthesized on the lagging strand are usually between 1,000 to 2,000 base pairs. So they're quite long. Don't think of them to be small, short things. they are quite long. Okay, so then what I'm trying to say here is that there will be a primer here, a short stretch of over 1,000 to 2,000. It will stop. Again, there will be another primer until this whole stretch is synthesized. And what I mean by that is that the primer will be here. The other primer won't be this side. It has to be up front, isn't it? Because synthesis is going that direction. So at this point, we have identified that on this strand A, synthesis will be lagging, discontinuous. So meaning this will be the lagging strand. It is the one that's going to have those Okazaki fragments, the short fragments of the DNA that are synthesized discontinuously using multiple primers. Let's look at strand B. Strand B has a 3' end. On this end, it is 5'. Can I synthesize DNA starting from this position? Yes, I can, because synthesis must be from the 5 to 3' direction. So this guy will only need one primer at the beginning. So after my primer is synthesized, I can continue synthesis all the way in the direction of the replication fork. Because synthesis is in the same direction as a replication fork, this strand is referred to as the leading strand. At this point, let's move on. If this is clear, let's move on to this diagram. I know I've skipped this. I tried to jumble them, but I've arranged them like this to allow me to explain what we're looking at. So when we look at the replication bubble, right, and this is the origin of replication, you're going to find that you're going to have a combination of leading and lagging strand on the same strand. Let's take, for example, the strand on top. Okay, on the same strand on top, within this replication bubble, you're going to have on one end the lagging strand. On the other end, you're going to have the leading strand. And again, the bottom strand, the same blue strand, is synthesis of the daughter DNA molecule. On one end, you're going to have the lagging strand. On one end, you're going to have the leading strand. How is this so? You must stand in this position in the middle. You kind of like split the molecule if you want, okay? So the first part we looked at was this end, isn't it? So if we split this molecule here, I'm going to go to the blue strand. If I cut it here, this position here is going to be a 3' end only for this part. Why? Because this end is 5. If I cut it here, as in I separate them, it will mean this end will be 3'. The other end that is cut will be the 5', isn't it? So what I'm trying to say here is that when you look at DNA, in fact, this strand is wrongly labeled. Oh my goodness, we've got 5' there and the 5' here. I apologize for that. I think I need to correct this one. But we can see how this model is going for us to be able to replicate it. What I'm trying to say here is that I hope you can see some of these mistakes, because I've got 3' here and 3' there. Or maybe it was just switched. But anyway, the understanding here is that if I cut this molecule here, this end here should be the 3' end. And how do I confirm if it is a 3' end? The strand below must be synthesized continuously with only one primer in the 5 to 3. Because these two strands must be anti-parallel. Okay? If this is the 3' end, this end here should be the 5' end. If it is a 5' end, I can't synthesize my DNA in the same direction. So it means it has to be synthesized discontinuously. So meaning this end here should be 3'. So I can easily correct myself. This end should have been 3'. So that I can synthesize this guy in the opposite direction to this replication fork. So meaning that on this same strand, when you split the origin of replication, one end will be a leading strand, the other end will be a lagging strand. And the same goes for this other strand. If you split it here, it means that this end will be a 3' end. That end there should be a 5'. So this should be continuous. If this end is a 3', this end here should be a 5'. In short, at each replication bubble, and by bubble at this point we know the region that has been unwound and we have active replication, we're going to have two replication forks. Those replication forks are going in opposite directions. We can clearly see this fork is going in this direction, this fork is going in that direction. The origin was here. So this is where you stand to start unwinding. So as you unwind, obviously, both ropes will be opening in opposite direction. In addition to that, at each replication bubble, we're going to have two regions of a lagging strand or lagging strand synthesis. We also have two regions of leading strand synthesis. At each replication bubble, what do you have? Two replication forks, that is very clear. We're also going to have two regions of lagging strand synthesis and, of course, our Okazaki fragments having multiple primers. We're also going to have two regions of the leading strand, the one synthesized in the same direction as the replication forks. So what we see here is that DNA replication is bidirectional. Bidirectional because synthesis is taking place going this direction and synthesis is taking place going that direction. Sometimes DNA replication is referred to as bidirectional. Why bi? Bi means two. It means in this direction and also in the other direction. You must be very clear to understand that when you stand at this position, you're kind of splitting this DNA into two. So to know which one will be the leading strand and the lagging strand, you must pay attention to the parent strand. You follow it. If on this end, you have a 5' end. If I go this way and I cut it here, this end must be opposite. It must be 3, isn't it? And if I'm going to look at the other one, this end now becomes a 5, and this end becomes a 3'. If this is a 3', this should be 5, this should be 3. It's very easy for you to designate those, those numbers, even if you don't see the phosphate or the hydroxyl group on carbon number 3. So I hope this is clear. So these are just notes. I think DNA synthesis is bidirectional, and I think we understand what that means. The new DNA strand is synthesized in the 5 to 3' direction, and we know why this is so. This is because the polymerase enzyme that is adding these new nucleotides can only function in this direction. And also what this means is that because the polymerase has a specific orientation of synthesis, one strand is synthesized continuously. This strand is referred to as a leading strand and always synthesized towards a replication fork or in the same direction as the replication fork. To start synthesis on the leading strand, you only need one primer, a single primer. And the lagging strand, this is a strand at the replication fork that is synthesized in the opposite direction to the fork and is synthesized discontinuously, obviously. And the short fragments of DNA referred to as Okazaki fragment. What this means is that each Okazaki fragment has its own primer. So meaning that if I compare the leading strand and the lagging strand, the leading strand only has one primer, while the lagging strand has multiple primers. If someone has turned on their mic because I'm sharing my screen, so I don't even know whether someone is asking a question and things like that. But anyway, I'll continue. So I think this is all about the leading strand, lagging strand, and what we've looked at here is the process of DNA elongation. So there was initiation, then elongation. So we've looked at the features of elongation. Not that we should get confused, because most of the time students fail to navigate this replication bubble. But maybe this is because you're looking at too many things at once. To start with, don't even look at the new strands. Just look at the parental strands given to you and number them very well. If on this end is a 5' end, when I cut this guy here, this end must take the opposite to what is at the beginning. And I'll do the same thing for this guy. The moment you are able to number or designate the orientation of these strands, it's easy for me to pick what should be on the inside. Because I know if on this end I have a 3' end, this end should be 5. This should just be opposite. And then I know if this side is opposite, then the strand will be discontinuous. So what I'm trying to encourage you here is that you when you're in your own free time, go and just practice how to designate or number a replication bubble. How do you designate this orientation of the slides? That's what I'm trying to say. And this is just for you to go and look at in your own time. Of course, most of that stuff has been hidden, But this is just for your practice. When you look, you should always stand at this position and cut this molecule. And in fact, the good part about this is that you haven't even been given what these ends should be. But I'm sure by looking at this synthesis, I can tell what this end should be, even if I haven't been given. And I hope you guys are able to do that. For now, it's something that you're going to do in your own free time. You should inspect this, even if you haven't been told what the orientation of this strand A is and B. Just by looking at this synthesis, I can tell, because I know the polymerase synthesizes in the 5 to 3 prime direction. So if this guy is a 5 prime end here, this end here should be 3, right? Anyway, what I'm trying to say here is that we need to be able to number these or not denote the orientation just by looking at what is going on, just by focusing a little bit. So you can do this on your own. If you fail, you can let me know, but I'm sure you're going to do it. Okay, so that was okay. So now, DNA replication I mentioned is not immune to mistakes. You know, there are accidents that can happen. And by that I mean, where there's, if the parent molecule was carrying thymine, instead of adding adenine, you add, let's say, guanine or anything else, or cytosine. That's a mistake, because that information is not going to be the same. It has changed. And that is referred to as mutations. I'm not sure we're going to look at mutations in this part of the course, but that introduces a mutation. And the language, the moment you change even just one nucleotide, sometimes you've changed the message. It's like you want to spell my name, and then instead of putting an SA, you put SB. It will be something else. So think of it like that. When we're saying DNA replication is not immune to mistakes, the mistakes we're talking about is introducing a mismatch. And by mismatch, I mean if there is a G, the daughter strand must have a C. The moment you put an A, it's something else. If there was a T on the parent, there must be an A on the daughter. The moment you put a mismatch, you've introduced a mistake. And these usually happen in DNA replication during elongation. But then the cell itself or the machinery has a mechanism to correct this. And what does it do? It uses the same enzyme. Of course, this time around, I'll talk about the others. It will use the same enzyme that is adding new nucleotides to correct. So in short, I'm trying to say DNA polymerase 3 can proofread the strand that it is synthesizing. DNA polymerase 3 has a proofreading ability to correct its own mistakes. Synthesis is in the 5 to 3, but to correct a mismatch, it uses its 3 to 5 prime exonuclease activity. What I'm trying to say here is that we should be very clear. For synthesis, you must be moving from the 5 prime going to the 3 prime. Now, when DNA polymerase 3 makes a mistake, it can correct its own mistake by using another activity, not polymerization, because remember, polymerization is making something. So it is using its exonuclease activity. Nuclease is like digesting, you know. Now exo meaning it's doing it from the outside. So exonuclease activity. So DNA polymerase 3 has an ability to proofread itself to see whether it has made the correct match or it's a mismatch. When it creates a mismatch, it is utilizing its 3 to 5 prime exonuclease activity. Not 3 to 5 prime polymerase. Now you're not making here, you want to take out the mismatch. So you're trying to break. The nucleases usually destroy DNA. Okay, so you're trying to destroy that DNA, the mismatch that you've put, in this case, a nucleotide. So what you see in this diagram is the feature of DNA polymerase 3, which is the 3 to 5 prime exonuclease activity. What does 3 to 5 mean? It means you're moving from the 3 prime end going towards the 5 prime end, just in simple language. Okay, what does 3 to 5 prime end means? It means you're moving from the 3 to 5 prime. But in nucleases, in this case exo, you're trying to say you're removing the nucleotide on its 3 prime end, the one at the outside, because this is exo. Now let me go to this strand. Okay, this is our template, and by that I mean the odd strand that has been unwound. So let's take, for instance, there was, synthesis. Okay, this is the strand that is the new strand that has been made. Synthesis is going from the 5 to 3, so obviously it is going in this direction. So these are like nucleotides that are being added. When a wrong nucleotide is added, okay, the polymerase can move in this direction. As you can see with the arrow, it can move from the 3 to 5 prime to take out that nucleotide. If it added 3 of them, it will be moving in the 3 to 5 prime direction. And because it is destroying, having this nuclease activity, it is referred to as a 3 to 5 prime exonuclease activity. Let me repeat myself. Synthesis of nucleotides is always from the 5 to 3. What this means is that you're moving from, if you're standing here, you start at the 5 prime end and you're moving step by step going this way until you add all these nucleotides. So that is why we say DNA polymerase 3 has a 5 to 3 polymerase activity. Polymerization, like you're adding more stuff, you're building something. Now, sometimes it does make mistakes. When it makes a mistake, it can reverse, kind of moving in reverse. Instead of moving from the 5 to 3, it can now move in the opposite direction, 3 to 5. And it will be taking them out one at a time. So it's going to chop this one, just like that until whatever mistake has been incorporated is removed. And when that is done, it will continue again synthesizing in the 5 to 3. So here it's just like an important concept. DNA synthesis occurs in the 5 to 3 prime direction. Proofreading means you're trying to correct. You spell something, you realize it's a mistake, you rub. So in this case, it's like rubbing what has been mismatched. It's going to move in the 3 to 5 prime direction. In a picture, you must think of it as it is standing this here and it is moving in this opposite direction, 3 to 5. This direction is not being picked from the parental strand. It's a direction of the newly synthesized strand, 3 to 5. Okay. And I think the next slide is just trying to reinforce the same, you've got your DNA molecule here, and then you have a mismatch. Where there's a G, you add an A. No, there's supposed to be a C. Because of this mismatch has to be corrected. So instead of it continuing synthesis in the 5 to 3, it's going to move in reverse. Take out the A and replace it with a C. The taking out of the A, it is using its 3 to 5 prime exonuclease activity. To add the correct base pair, which is synthesis, it is using its 5 to 3 prime polymerase activity. Okay, now the primers that were used to initiate synthesis, remember I mentioned they are not DNA in nature. They are RNA. And when we look at our DNA, it's not a camouflage of DNA and RNA. No, it's just pure DNA. So we need to remove these primers. We don't need them. Remember, the only thing why they were brought into this equation is just for them to provide that, the hydroxyl group for us to start synthesis. So when replication is done, we need to take them out. So another enzyme comes in to help the cell take out these, primers. And what we should remember here is that the RNA primer is RNA in nature. It's not DNA in nature, no. So that is why it has to be removed. So to remove the RNA primer, DNA polymerase 1 comes in to remove these RNA primers. The primers that were used to synthesize DNA, one on the leading strand and multiple of them, even 1,000, depending on how long that thing is, are removed by DNA polymerase 1. Now, DNA polymerase 1, just like any other polymerase, has a 5 to 3 prime polymerase activity. It has, when I say polymerase activity, meaning it can synthesize DNA also. And the synthesis is going to follow the same direction as polymerase 1. So it can synthesize in the 5 to 3 prime direction. But when it comes in to remove this RNA primer, it is using its 5 to 3 prime exonuclease activity. Now, don't confuse these two. Remember, polymerase 3, the exonuclease activity is different. And this guy is actually very funny because it's among, maybe if not the only polymerase that's got this ability to move in the 5 to 3 prime exonuclease activity. Meaning that it will start chopping the primer from the 5' end. That's what this means. I have a slide where I'm going to show you this. The key message here is that 1, the primers that are on the DNA serving as the initial template must be removed. To remove them, there's another polymerase that does that. This polymerase is referred to as DNA polymerase 1. It removes these RNA primers. Now, to remove them, how is it doing it, or how is it moving? It is starting from the 5' end of that primer, moving to the 3'. And because it is removing, we can't say polymerase. We are saying it is exonuclease activity. So DNA polymerase 1 removes the RNA primer using its 5 to 3' exonuclease activity. This is opposite what DNA polymerase 3 has. DNA polymerase 3 moves in the opposite direction, 3 to 5, but this guy can move in the 5 to 3. But synthesis, when it removes the RNA primer, it can also synthesize DNA. And when synthesizing, it also uses the 5 to 3' polymerase activity. So these two can be a bit confusing, but I hope you can understand the movement of these polymerase in these activities. When you're taking out, it's exonuclease. Okay, like, killing, destroying something. The direction of movement on the new strand, or in this case, the primer, you start at the 5' end and you move in this direction. Once you remove the primer, there's going to be a gap. That gap needs to be filled. So it does not recruit DNA polymerase 3, no, because DNA polymerase 3 is a long-range, enzyme. It needs to synthesize continuously, not stopping anyhow. No, remember I said even the Okazaki fragments, they're quite long. They could be 2,000 base pairs, but these regions are very short. So the primer is very short, about 10 to 25 nucleotides. So it's quite short. So you don't want to engage an enzyme like polymerase 3 to do a job that can be done by a small enzyme. So polymerase 1 comes in to synthesize short DNA fragments. And the direction of synthesis in the same way is from the 5 to 3. And then the question you might wonder, why doesn't it need a primer? No, because remember there's already an existing DNA. It's just been chipped. So it can easily synthesize the part that has been removed. Now, once DNA polymerase 1 has synthesized the short primer, it does not connect, it kind of, the DNA molecule remains with the nick, like a small portion where there's no phosphodiester bond. And that region of the backbone needs to be ligated or connected to join the backbone of the DNA that has been synthesized after removing the primer, replacing the DNA. The enzyme DNA ligase comes in to seal the nick, the small portion. And the nick here is not adding, DNA ligase does not add nucleotides, no. The nucleotides on the region where there was RNA primer are added by DNA polymerase 1. But there's a challenge here. DNA polymerase 1, while it can add, nucleotides on the region where there was a primer, it can't form the bond to connect, phosphodiester backbone, the sugar phosphate backbone. So to do that, to seal the sugar phosphate backbone, another enzyme comes in. This is referred to as DNA ligase. Getting is putting things together. Okay. Yeah, so sometimes this is also referred to as nick translation. Now, I just want you to look at this slide to understand what 3 to 5 means or 5 to 3 means. So, I remember saying that DNA polymerase 3, to correct itself because it has got this proofreading ability and it can make mistake, it acknowledges that. It moves in the 3 to 5' exonuclease because you're destroying something, or destroying nucleotides in this case. So what this means is that you're moving from the 3' going this end. That's what it means. 3 to 5. If this is a strand of DNA, you must stand at the 3' end and start moving in this direction. DNA polymerase 1, it also has an exonuclease activity. But instead of starting from the 3' end, it stands at the 5' end and moves in that direction. But both of them to synthesize, they move in the 5 to 3. So this one just requires you to have an understanding of the orientation, or should I say the polarity of DNA. When an enzyme is said to have a 3 to 5', what are you trying to say? You must stand at the 3' end and move towards the 5. When an enzyme has a 5 to 3' exonuclease activity, you can't stand at the 3'. Remember, you've been guided. It is 5' to 3, meaning you start from the 5' end going in that direction. And this is just a summary of what DNA polymerase 1 is doing. I think you first of all, the primer is elongated by DNA polymerase 3. By this one means it's when you synthesize the primer, the other part is done by DNA polymerase 3. The primer has to be removed. It is removed by DNA polymerase 1. Once you remove the primer, you create a gap. That gap has got the base pairs on the parent molecule, but it needs now nucleotides that are DNA in nature. So DNA polymerase 1 can do that, and it does the synthesis, but it cannot fix the sugar phosphate backbone. It kind of leaves a nick. So to seal the sugar phosphate backbone, the enzyme DNA ligase comes in to just seal that position. So the completion of elongation, what you have there is the removal of these primers. We don't need them. When we remove them using DNA polymerase 1, we need to replace that region where there was the RNA primer with DNA, with nucleotides. This is done by DNA polymerase 1. One of the challenges of DNA polymerase 1, it can't seal the sugar phosphate backbone. So what happens here? Another enzyme is loaded. This is DNA ligase to just seal the sugar phosphate backbone. So I want to emphasize, DNA ligase does not add nucleotides. No. Its job or its role is just to... Yes?
Sorry, madam, to interrupt. We've been learning for about 4 hours, so the people were kind of asking if we could just end here.
Okay, you know what? I'm actually about to finish. In fact, I wanted to ask, because tomorrow I wanted to see if I can have a repeat lecture for those who have not been able to join. In fact, I wanted to discuss with Mike, because I'm supposed to start translation. And right now, how many slides do I have left? Let me just show you. It's not a long lecture, so I wanted to end here. So this is just a repetition. So
some of us, madam, will be having a class at 18, so we need to charge our phones.
Please speak up.
I'm saying that some of us need to charge our phones. We'll be having class at 13 hours again.
What time is it? It's 12. Well, that's up to you, because tomorrow I wanted to just have a makeup lecture. I don't know what others think, because the notes are with you. I'm about to end in four slides, and I'm supposed to start translation.
Madam, I think we can end here, because there's a guy who does YouTube live streams. So the lectures that we have in biology, most of them are on YouTube.
Okay.
So maybe we can end here, then tomorrow we'll continue from here.
Okay, so what I'm asking is that I want to acknowledge the fact that not everyone is able to join. So what I wanted to do for the Wednesday lecture is to have a makeup lecture for those who haven't been able to join. That's what I wanted to say.
Yes, madam, that's actually a good idea. I think it's going to help those that have missed the lectures to catch up.
Thank you very much. So tomorrow I'm having a repeat. I'm going to ask those of us who've been able to join the lectures on DNA replication, allow those who haven't been able to join tomorrow at 8 hours. So for us, we can end here. I think we can continue next week, but for tomorrow, I think if they can even allow me 2 hours, I'd want to have a makeup lecture for those who haven't been able to join. Please do not join. Thank you very much, and have a good day. So there's a question, do I stream? What does that mean? I don't know what that means. Maybe Mike can guide me. Someone was asking whether he can stream both lectures. But maybe Mike, we can talk on the side so people can go and rest and go for another lecture. Thank you very much, guys. Take care. See you Monday.